I'm very excited about interdisciplinary research in general. I'm personally trained as a physicist, and I got rid of biology in 10th grade in school. So the excitement for me is that I learned so many new things talking with people from different disciplines.
You've developed DNA paint, a microscopy technique that allows us to see individual molecules inside cells. Could you explain what problem this technology solves that traditional microscopy cannot?
So a little bit more than 10 years ago, super resolution microscopy got a Nobel Prize in chemistry to beat the diffraction barrier. So DNA paint is a technique out of the zoo of super resolution microscopy techniques, and what we do differently, we use programmable DNA molecules to label biomolecules in cells and read out their position and identity by complementary strands. So the problem in super resolution microscopy that DNA paint solves is multiplexing and higher spatial resolution, basically.
So with DNA paint and its many iterations, you've really pushed the boundaries of both resolution and throughput. What are you excited about for future developments of this method?
So I think we are traditionally a technology development lab, and I'm super excited about new tech in general. But I think the technique is at the point where you can now apply it to answer fundamental basic biology questions, and eventually also push it into more translational areas where you can use it in applied methods, for example. So one thing I'm really excited about, to actually apply this in a more biomedical context in the future.
And any other methods that aren't so DNA paint based that you're excited about? Or
I'm excited about many things. I'm generally excited about spatial biology at this point. So putting sort of high content information into spatial context in biological systems. And I think I'm excited to see a lot more developments in that direction and a lot more insights into biological systems in the future.
As a sort of related follow-up question, one thing we think about a lot is that methods are really only as good as the discoveries that they enable. So what avenues of research applying these techniques are you most excited about?
So I think one thing, also a study that we recently have done with DNA paint, is that we looked at the molecular mode of action of monoclonal antibody drugs in cancer therapy. And I was actually astonished that for many of the drugs that are also FDA approved since decades, the molecular mode of action, so how they actually work, interact with cellular systems, is pretty much unknown. So you know that they have a high efficacy and they are safe, right? But the molecular mode of action, how they work or how they don't work, and when they fail, has not been known. And so we teamed up with a team with Roche in Switzerland to look at the molecular mode of action of some of those B-cell derived cancer monoclonal antibodies, and could see that their functional outcome is related to the way that they are inducing clustering in molecules on the cell surface. So we could link function with structure or organizational capabilities of these molecules. And I think that, this will not solve health and disease by itself, but I think it could maybe give a little bit of an input on how you might develop drugs differently in the future using these high-resolution microscopy techniques.
Your work bridges physics, chemistry, and biology. What can you tell us about the value and challenges of working at the intersection of disciplines?
So I'm very excited about interdisciplinary research in general. I'm personally trained as a physicist, and I got rid of biology in 10th grade in school. So the excitement for me is that I learned so many new things talking with people from different disciplines that gets me excited to learn new things every day. So the challenge that comes with this is translating between those different disciplines, right? So if you talk to a computer scientist, their way of thinking about a problem is very different from a physicist or a biologist or a chemist. And I think this is a challenge, but it's also the opportunity to bring together different fields to work on a problem that everybody by their same discipline would not be able to address.
Okay, and the challenges?
Challenges is translating between the different fields, right? So, yeah.